Good afternoon. To begin with, I want to thank the organizers. It's really a pleasure and honor to be here today. The topic I was asked to discuss with you is the one you can read there, Carcinogenesis and Molecular Events in Gastric Cancer. This is a bit ambitious for half an hour. I'll see what I can do with you. And what I propose myself to discuss is to have gastric carcinoma as the end point, the end product of the interplay between different factors, which are the environment, host susceptibility, and the molecular events, which are somatic events. So I will address briefly gastric carcinogenesis, the classic model of Correa, the molecular features of different types of gastric carcinoma, briefly the hereditary gastric carcinoma, coming up to an overall view of gastric cancer these days. If you begin by gastric carcinogenesis, this is the famous Correa model, in which you know that gastric cancer is the end product of a sequence of events that by morphology encompass superficial gastritis, chronic inflammation, it means superficial gastritis, atrophic gastritis, intestinal metaplasia, dysplasia, and carcinoma. All of you are aware of this. However, there is in this model some things which are new and which I would like to emphasize. First, from etiology, to emphasize that it's not H. pylori but other factors that are important. From the morphology and the possibility of reverting the lesions and avoiding gastric cancer by treating H. pylori infection, to tell you that evidence from literature clearly show that this is only efficient if the patients are treated when they still do not have precursors of gastric cancer, otherwise it will be too late. Just to mention the role of the bone marrow-derived cells, as demonstrated by Jean-Marie Haug, we do not have time to address this. But what I would like to emphasize also is that in this scheme, you can see not only intestinal metaplasia, which is intestinal metaplasia in which gastric mucosa, you have intestinal phenotype with intestinal markers, but also something that has been designated as SPAM, and that is getting huge importance. What is SPAM? SPAM is spasmolytic polypeptide expressing metaplasia, is a type of change in gastric mucosa in which you have the expression of 3 foreign peptides. I will come back to this later because I think this is very important. So, and taking this as a background, what I will briefly address now is the importance of H. pylori. All of us are well aware of the importance of H. pylori as one of the most important causes of gastric cancer, taking into consideration the environmental factors, not forgetting that there are other environmental factors that are also important, and also that the host is also a very important factor for the development of gastric cancer. Here you have the cascade of events, and what I would like to emphasize now is that the progression along this cascade results from the interplay of H. pylori itself by the products secreted by the bacterium, CAG-A, VAC-A, also from other factors from the environment, and also from the host polymorphisms that I'll briefly address. And if you think about H. pylori, you should be aware that the relationship between H. pylori and the host is not so passive as you could imagine. H. pylori has 2 adzines, which are very important for the establishment of a relationship with gastric mucosa, because one of these adzines, designated by BAP-A, and this comes from blood group antigen binding adzine, is an adzine that binds in the gastric mucosa to blood group antigens H type 1 and Lewis B. H. pylori has another adzine, which is designated as SAB. It comes from sialic acid binding adzine, and this adzine binds in the gastric mucosa to sialylated antigens, namely sialyl-Lewis A and sialyl-Lewis X, which normally are not expressed in gastric mucosa. And what is relevant in the relationship between H. pylori and the gastric mucosa is that the bacteria itself can induce in the gastric mucosa the de novo expression of these sialylated antigens, namely sialyl-Lewis X, which contributes to a long-lasting colonization of gastric mucosa by H. pylori. From the other side of the spectrum, we were discussing environment so far and have not detailed the role of other factors from the environment. From the host itself, there is something which is relevant in this interrelationship, and this is related to polymorphisms, specifically polymorphisms of the genes that encode for pro-inflammatory factors. And just to give you an idea of the relevance of this, if you take in consideration the risk of gastric cancer development only on the basis of the virulent factors of H. pylori, VAC-A, and CAG-A, if present, these virulent factors increase the risk of gastric cancer something like 15 to 17 times. If you consider only the polymorphisms of the host in what is related to the expression of IL-1 gene, this increases the risk of gastric cancer something like 3.3. For the individuals who are colonized by these virulent strains of H. pylori and also have the polymorphism giving susceptibility for the development, those who have together these two factors, the risk of development gastric cancer increased to something which is 87. And this is something we cannot forget when you think about the pathogenesis of gastric cancer. So what I was discussing before was the gastric carcinogenesis, and what I'm moving now is the molecular features of the different types of gastric carcinoma. Just to highlight that when you think of gastric cancer, you should take in consideration two different levels. One is sporadic cancer, the one that exists the most frequent, accounting for about 90% of the cases, and familial hereditary cancer that contributes to about something 10%, and the hereditary only 1%. And to discuss the features, the molecular features, you have to have in mind what is the classification of gastric cancer. This is the classification proposed by the WHO. We have discussed yesterday, some of you may have heard yesterday, the new classification in which you have papillary, tubular, mucinous, poorly cohesive carcinoma corresponding to the signet ring cell carcinoma in the previous classification, and the mixed carcinomas. And you can say, roughly, that the tubular-papillary carcinoma, according to WHO classification, corresponds to the so-called intestinal type of carcinoma, according to Lorraine classification. And you can also say that the poorly cohesive type corresponds largely to the diffuse carcinoma, according to Lorraine's classification. The profile of these two entities is different, not only regarding epidemiology, but also biology of the disease. These tumors with glandular structure tend to give rise to blood-borne metastases, while those constituted by poorly cohesive cells tend to disseminate to the peritoneum. And if you think of the molecular changes occurring in each of these morphological types, there is a huge amount. No time to discuss them in detail. I will briefly refer only to the major changes that can be used in routine practice, and they are related to the expression to this disturbance of HER2, epidermal growth factor receptor, microsatellite instability, and E-cadherin gene, also designated by CDH1. I'll begin by CDH1 gene. As you all know, CDH1 gene encodes for E-cadherin. E-cadherin is a cell adhesion molecule, and in the sporadic gastric cancer, there are mutations occurring in about 50 to 70% of the cases in this gene. These mutations are concentrating the central part of the gene, and there is here an hot spot in exons 8 and 9. The consequences of these mutations, when there is an activation of the second allele, and the inactivation of this second allele generally occurs due to promoter methylation, there is loss of expression of E-cadherin in diffuse gastric cancer. This is the best example of the involvement of a gene in the development of a phenotype. But E-cadherin is involved not only in the diffuse type of gastric cancer, is also responsible for the development of the so-called mixed gastric carcinoma, which the two components of these tumors have the same chromosomal alterations, but there is a major change, which is the loss of expression of E-cadherin in the component of the tumor constituted by poorly cohesive cells, which is due to the mutations of E-cadherin gene. So E-cadherin gene is also responsible for the development of this specific phenotype in gastric cancer, and we think that these mutations are the basis of these phenotypic divergence. Further, if you think about the E-cadherin gene, we know these days, and this is data produced recently by our group, that E-cadherin can be downregulated in gastric cancer of the intestinal type, and this is due not to mutations as it occurred in the previous cases, but now is due to the disturbance of the regulation of microRNA, specifically microRNA 101. And the lack of this type of microRNA causes functional alterations of E-cadherin with downregulation of E-cadherin in intestinal type of gastric cancer that can contribute to the progression of disease. So all the E-cadherin gene alterations is linked mostly with diffuse type of gastric cancer. We know now that the alterations can be behind also mixed and intestinal type. The other type of marker in gastric cancer is the HER2 gene, and all of us have discussed this several times during this meeting. You know that HER2 is an oncogene. The change in gastric cancer, mainly intestinal type, is amplification in this gene. It's disputable if it is a prognostic factor. Data in literature are controversial, but there is a relationship between the amplification and the occurrence of blood-borne metastasis. And the relevance of this change, all of us are aware, is related to the probability or the possibility, which has been demonstrated in TOGA trial, of using HER2 as a target of a therapy with trastuzumab, and you are much well aware of this than me. If you move now to microsatellite instability, all of you know that microsatellite instability is a marker in colorectal cancer of tumors which have microsatellite instability and have a very different profile and behavior from those which do not have. And the same profile occurs in the stomach because the tumors with microsatellite instability, they do have a best prognosis as it occurs, a better prognosis than the others without microsatellite instability as occurs in the colon. And this is an independent prognostic factor and is a marker of good prognosis. We can state not yet used in the stomach as far as I'm aware for therapy, and we know that in the stomach is mainly due to the methylation of HMLH1, as it happens also in the sporadic cancers in the colon with microsatellite instability. So there is here some similitude between the tumors with microsatellite instability in the stomach and in the colon and rectum. It's curious when you search in the stomach for alterations in KRAS and BRAF, and when we search specifically for mutations, and that's what I'm talking about now, you find mutations of KRAS in the stomach only in the tumors that have microsatellite instability, and this can be of some relevance regarding prognosis and treatment. But this is far behind what we know these days, and is coming for the new technologies from high-throughput technologies, encompassing, and if you go to literature, there are dozens of papers published recently about different alterations in gastric cancer. It's impossible to go through all of them, but I think that literature has now evidence, some evidence that deserves to be discussed with you today. The first I would like to refer is this one published by Dr. Shah, who has been here yesterday and who presented these results last year in this meeting, in which his group was able to identify in gastric cancer 3 different groups, which they designate as proximal non-diffuse, diffuse, and distal non-diffuse. And they announced by that time that further studies were being performed in order to better elucidate this, which I'm not aware of. If Dr. Shah is there, most probably will participate in the discussion. A very relevant study that has been recently published, this was 2012, by the group of Patrick Tan in Singapore, demonstrated in gastric cancer for the first time something which is very important, and this thing is that in gastric carcinoma, you can get amplification of different genes Not only HER2 we knew already of, so HER2 or HER-BB2, if you prefer, is amplified in gastric cancer, but this study clearly demonstrated that there are other genes that are also amplified in gastric cancer, such as fibroblastic growth factor receptor, KRAS, epidermal growth factor receptor, and MET. What's the relevance is? The relevance is that if these genes are amplified, they may become good targets for therapy, as it happens already with the target therapy directed to HER2 overexpression due to amplification. The additional issue is that KRAS amplification is associated with a poor prognosis in patients with gastric cancer. This was not known before. In our experience, we had tests for KRAS mutations, but we had not been able to identify these amplifications, so this is new knowledge should be taken in consideration. Another paper which is extremely important is this one, again coming from the Singapore group, from Patrick Tam, and published in 2011 in Gastroenterology, in which these authors were able to identify two genetic profiles in gastric cancer, which were identified on base of cell lines. They designate these profiles as intestinal and diffuse, and they were able to show in primary tumor samples from two different series of cases that there was a correlation between this genotype and the morphology of the tumors. It was not perfect, but was making sense. In a way, it was telling us that what we see by morphology, the glandular tumors, intestinal type, they have behind the genetic profile, which is different from the tumors with the diffuse morphology. So it was making sense. And they claimed also that this could have implications for the evaluation of the prognosis of the patients, the diffuse type of cases having a worse prognosis. So they claimed that there were intrinsic subtypes of gastric cancer based on the expression pattern of the tumors. But even more relevant, and this is something I want to discuss with you in detail because will lead us to revisit gastric carcinogenesis, is this recent paper is in the PubMed, ahead of print. This was published online in 2013 and is coming also from the Singapore group. And what these authors have shown by consensus hierarchical clustering is that it is possible to identify in gastric cancer 3 different types. The authors designated, by reasons that I explained, these types as mesenchymal, proliferative, and metabolic. Is this relevant? What was the basis of this? The basis was that in the mesenchymal type of tumor, the genes that are alterated are genes mainly related with cell adherence and relationship with extracellular matrix. I'll detail a bit later. And the proliferative type, the genes involved, amplified, are most related with cell cycle and DNA replication. And for the metabolic type, which is the smallest group, the genes that are altered by amplification or overexpression are genes that are normally expressed in gastric mucosa. And the authors decide to designate this group as metabolic because several of these genes are genes that, according to the annotation systems, 2 different ones, are genes involved in various metabolic processes in gastric mucosa. Why is this relevant? If you put this evidence now with morphology and other features of gastric cancer, what the authors have seen is that for the mesenchymal type, genotype, there is a correlation with diffuse type of gastric cancer. There is a correlation between the proliferative genotype with the intestinal type of gastric cancer. And for the metabolic, this was observed both intestinal and diffuse morphological types. There was a relationship between the genotypes and copy number alterations. The mesenchymal genotype correlated with low number of copy number alterations. The proliferative phenotype correlated with high copy number changes, telling you that this is the type of tumors which have chromosomal instability and making sense with the proliferative and the activation of cell cycle genes and DNA replication genes. And P53 mutations were searched also and were mainly identified in the proliferative type, which has a number of changes of copy number alterations, and in which you detect mainly hypomethylation. What was also shown in this study, and this is relevant for us pathologists, is the different types of genes that are altered. If you look at the mesenchymal type, which corresponds to the diffuse, major genes are involved in epithelial mesenchymal transition and are genes which are related to cancer cells, stem cells, which are important in this specific type of tumor, which is the diffuse type. If you look at the proliferative, with high number of copy number alterations, with P53 mutations, there are other genes that are altered, such as these transcription factors, MET and KRAS. And if you look now to the metabolic, the important thing is that one of the most important genes is TTF2. What is TTF2? Why is this so relevant? What I'm now highlighting is that the metabolic pattern of genotype is the one that is, in a way, requiring a good interpretation. And according to these authors, this is relevant because the tumors with the metabolic genotype, they are sensitive to 5-FU. So from this, you could get information for the treatment of your patients. According to this study also, the tumors of the mesenchymal type are sensitive to mTOR inhibitors, so this could be relevant also. And I come again to the SPAM metaplasia and to the overexpression of TTF2. What is TTF2? TTF2 is a trefoil peptide, is expressed in the stomach, is co-expressed with mucins. One of the trefoil peptides is expressed in the stomach with MUC5AC and the other with MUC6, and these are markers of gastric differentiation. And in the stomach, we know now, and we have provided much of this information in our group, together with other groups, namely recently from Japan, that in gastric cancer, even those with a glandular structure that according to Laurian classification we would call intestinal type of gastric cancer, this is an incorrect terminology if you take in consideration the differentiation at the cell type. Because some of the tumors, they have a gastric phenotype, and some others, they have intestinal phenotype. So this, in a way, stimulated us to revisit gastric carcinogenesis and to go back where to? To go back to dysplasia, which is the immediate precursor of gastric cancer with glandular structure. We went back, and we know by morphology, according to WHO classification, that there is also by morphology two types of dysplasia in the stomach. One which is the intestinal type, reminding you of what you see in polyps in the colon, and the other is the gastric phenotype, which is distant and is a bit difficult, let me tell you, for a pathologist to recognize. But we have the tools now to characterize better these types of this differentiation using the immunophenotype. And actually, we have demonstrated that there are some types of dysplasia in the stomach in which you only have the expression of MUC2 or CD10, which are markers of intestinal differentiation, and you do not have the expression of MUC5AC only observed in normal gastric mucosa and is totally shut down in dysplastic lesions. So this is dysplasia of the intestinal phenotype. Let me remind you again that there are a couple of there's a percentage of gastric cancers in which the differentiation is also with the expression of MUC2 and CD10. So this type of dysplasia would be a precursor of a gastric cancer, gastric carcinoma of intestinal phenotype. On the contrary, there is in the stomach some dysplastic lesions, a proportion of those, in which what we observe is the overexpression of MUC5AC, which is co-expressed in the stomach with the trefoil peptide, remember of the metabolic pathway of gastric carcinogenesis, and together with MUC6, which is also expressed with the trefoil peptide, and together this demonstrates that in the stomach, this type of dysplasia, without the expression of MUC2 or CD10, it is what we call these days dysplasia with gastric foveolar immunophenotype. So what's the relevance of this? According to the evidence we have recently gathered, this is quite relevant because if you look at immunophenotype and if you look at gastric phenotype, the large majority of those are situations in which you have high-grade dysplasia, while the large majority of these plastic lesions with intestinal phenotype, they only have low-grade dysplasia. On the top, in our experience, for cases with high-grade dysplasia with gastric phenotype, there was coexistence of gastric intermucosal carcinoma in 8 cases. So in our minds, this is relevant for the understanding of gastric carcinogenesis. And putting together all that I've said, what we think these days is that in gastric mucosa, you have superficial gastritis, you have chronic atrophic gastritis, we have H. pylori and other factors, and you can have either intestinal metaplasia evolving along displaced intestinal phenotype to adenocarcinoma intestinal phenotype. On the other side, you can have SPEM metaplasia with dysplasia of gastric type and adenocarcinoma of the gastric phenotype. And what we would really like now to do, and we have challenged already Patrick Thum, is to demonstrate that there is a relationship between this gastric phenotype of gastric adenocarcinoma with the metabolic phenotype it suggests, and this is relevant because of the consequence for therapy. We move now very briefly to hereditary cancer, and just to mention what I summarized yesterday, that we have 2 types of hereditary cancer in the stomach, the so-called diffuse, which involves thousands, hundreds of foci in the stomach, for which we know the precursor lesions, for which we know the genetic changes affecting E-cadherin gene, mutations occurring along the whole length of the gene, for which we know the impact of these mutations, most of them being truncating mutations, and patients carrying these mutations should be submitted to prophylactic gastrectomy. The other type of gastric cancer of hereditary type is the so-called GAPS. This is the gastric cancer. The tumors have a glandular structure which develop in the setting of the proximal polyposis of the stomach. So we could say, if we would make an analogy with the Lorentz classification, this would be a type of hereditary intestinal cancer in the stomach. So this brings me back to the initial slide and to the conclusions and to a tentative to bring to you a global picture of what happens in the stomach regarding gastric carcinogenesis that is actually much more complex than what is behind the Correa model. So in the sporadic gastric cancer, you have a pathway leading to diffuse carcinoma. You have another pathway leading to carcinoma with glandular structure, which can have an intestinal or a gastric differentiation. And you have the hereditary cancer, one of the types of the diffuse type, the other of the types, the glandular or intestinal type, if you prefer. So this is a bit more complex, but I think this gives you a comprehensive interpretation of what gastric carcinogenesis seems today. Is this relevant? I think this is very relevant these days for what I would like to designate as personalizing pathology in analogy with your personalized medicine. This is something from an interview that I was asked to give and from which I bring what I think is the most important. And the most important is that we pathologists, put together all the information from the molecular studies for the better understanding of the diseases. And it is today absolutely mandatory that we pathologists work together with you oncologists and with geneticists in a perfectly synchronized way for the benefit of the patients. Because I think these days we are approaching next generation sequencing pathology. There is a huge number of people behind the evidence I've shown to you. We have done all these studies in collaboration with many centers in the Netherlands, in the United States, in Canada, in UK, at the Mayo Clinic in the United States, in other localizations, also in Australia and New Zealand, and this has been the source of a huge pleasure. And this is all, and I want to thank you so much for your attention.